Hello and good morning, everyone. Thank you so much for joining this presentation. I'm Shikshya Gautam, a PhD student from Bowling Green State University, and today I will be presenting my research on the ecological and health risk assessment of potential heavy metal contamination in crops grown in soil amended with dredged material from the western Lake Erie Basin, Toledo Harbor, Ohio. From the previous presentation from Dr. Nilika, we learned about how the tons of dredged sediments are excavated each year from the Toledo Harbor, and after the ban of open water disposal, it has been stored in confined disposal facility. Since then, different environmental agencies and organizations are working on to find the beneficial use of dredged sediments. So dredged sediments has various uses, and in our lab we focus on the agricultural use and found that it meets the optimal value required for the various soil parameters and has the potential to be used as farm soil amendment. We also learned about the effect of dredged sediments on soil and crop health from our previous presentations. My current research will focus on to examine the potential ecological and human health risks associated with heavy metal bioaccumulations in the crops grown in soil amended with the dredged sediments. To address this, I have two research objectives. One, to determine the potential bioaccumulation of heavy metal contaminations of the farm soil amended with different ratios of dredge. Second, to determine the ecological risk coefficient, transfer factor, daily intake of metal, health risk index, and their implications in soil and human health. Here is an overview of my study design. I had three organically managed fields with different ratios of dredged material. Plot 1 received 40 tons per acre of dredged material. Plot 2 did not receive dredged material. And plot 3 received 80 tons per acre of dredged material. Fields were divided into the grid by using Google Map software, and GPS points were located for each grid. The soil samples were collected at time zero and after harvesting both corn and soybean. In year 1, corn was grown, followed by soybean in year 2. My study site is situated at the Agricultural Incubation Foundation, which is about 10 miles from Bowling Green State University. These rectangular boxes in the first picture represents my three different fields with different dredge ratio. Images here in the right show the dredged sediment in the field and how they were spread into the field using these equipments. These are the same equipments that farmers usually use to spread manures in the field. This slide gives an overview on the crop yield data. In the year 1, when corn was grown, the yield was lower in the field which received the dredged material. However, the yield increased in the next year when soybean was grown. We realize it's a good practice to spread dredge during summer as we spread dredge material in the spring, and the dredge material as well as the soil was moist, and then we used a heavy equipment to spread the dredge material. Therefore, the compaction may have affected the yield in year 1. Taking this into consideration, Dr. Angelica is working on another project where we will be studying the how traffic affects the yield, as we also saw in the previous presentation by her. This slide shows the overall method for my research. However, for today's presentation, I will be focusing on the heavy metal analysis. So for my soil and biomass samples, I first ran the ecologia digestion, followed by using ICP-OES, which is inductively coupled plasma optical emission spectroscopy. It is an analytical technique to detect and quantify the elements in liquid samples. It involves introducing the samples into the plasma torch, where high temperature cause elements to excite and emit characteristic wavelength of light. The emitted light is measured by detector, allowing for them to determination of each element's concentration based on the spectral lines. After receiving concentration data, I further calculated the safety indices. And these are the four safety indices that I will be focusing on. I have calculated the ecological risk assessment and potential ecological risk of heavy metal. I will be calculating transfer factor of translocation, daily intake of metal, and health risk index. These indices helps us to understand and evaluate the potential health risk and environmental risk associated with the heavy metal contaminations. The ecological risk assessment tells us about the degree of contamination of metals with respect to the background levels. For this, we calculated accumulation coefficient, which is the ratio between the concentration of metals in the soil from different treatment to the concentration of metals in the background level. If the accumulation coefficient is greater than 1, then the metal concentration is more than the background level, thereby enriched. If the accumulation coefficient is less than 1, then the metal concentration is less than the background level, thereby depleted. If the accumulation coefficient is equal to 1, there is no difference. With this accumulation coefficient, we further calculated the potential ecological risk. We calculated the potential ecological risk by multiplying the accumulation coefficient with toxic response factor. Uh, and this factor represents how sensitive a bioorganism is to it. And this toxic response factor for certain elements are uh, fixed according to the study conducted by Nata Kiru Timana. And then after calculating these values, we then compare the values to this table to see the ecological pollution degree. If the potential ecological risk is less than 40, there is low ecological risk. If the value is in between 40 to 80, then there is a moderate ecological risk. If the potential ecological risk coefficient is between 80 and 160, there is a considerable ecological risk. And if it's between 160 to 130, 160 to 320, there is a very high ecological risk. The next important index is the transfer factor of translocation. This is the ratio of metal concentration in plants to the metal concentrations in soil. A transfer factor is greater than 1 indicates an increased chance of metal translocation from root to the aerial parts of the plant, which is significant for accessing the risks of human exposure through crop consumption. Finally, we access the daily intake of metal and then health risk index. This daily intake of metals helps us understand the potential health risk. The daily intake of metal is calculated where by using this formula where Cm is the metal concentration in the crop, Cf is the conversion factor, and DFI is the daily intake of the crop, which is BAW is the average body weight of individual. After calculating the daily intake of metal, we further calculated health risk index, which is the ratio of daily intake of metal to the reference oral dose recommended by US FDA. If the health risk index is greater than 1, the human population is at risk to eat the crops grown in waste material. If it is less than 1, the exposure level is considerably safe. This indices provides a comprehensive view of potential risk and allow for better management and mitigation of heavy metal contamination in the environment. These are the pictures that I took while I was working in the lab. In the first picture, I'm preparing a Courageous solution. The picture on the right is ICP OES equipped with the auto sampler. Coming to the result, these two graphs shows the ecological risk assessment. Values calculated from the soil where corn and soybean were grown in different treatments. Values higher than 1 represents the metal concentration is more than the background level, thereby enriched. Value less than 1 represents the metal concentration is less than the background level, thereby depleted. The background reference in this case was the farm soil, which did not receive the dredge treatments, which is 0 dm tons per acre. As you can see, in the x-axis we have different ratios of dredge sediments, and in the y-axis we have accumulation coefficient. Ecological risk assessment values for both corn and soybean has a similar pattern for both 40 tons per acre and 80 tons per acre. The values for the most of the element is less than 1 or close to 1. The arsenic has slightly higher value than other elements in 40 tons per acre. However, in the 80 tons per acre, the ecological risk assessment value for arsenic decrease, showing adding more risk can dilute the metal concentration. This table presents us the ecological risk coefficient values for soil where corn and soybean was grown. If we compare the values with this ecological pollution degree from this table, all of the elements have values less than 40, representing low ecological risk. Here also the arsenic has the higher values among all elements and shows the similar pattern of decreased value in 80 tons per acre than 40 tons per acre. Similar study was conducted in our lab for specialty crops by Olusola Oyoyumi. The specialty crops were tomatoes, lettuce, and strawberries. It was a greenhouse experiment. He had 3 treatments: 100% soil, 100% raised material, and mixture of soil to raise in the ratio of 90:10. The ecological risk assessment values for most of the elements was below or close to 1 in the mixture, which is similar to our study. Arsenic and zinc was higher in the 100% raised material. The ecological risk coefficient for all the crops in all the treatments was less than 40, showing low ecological risk similar to our study. This graph shows the transfer factor of translocation from the study conducted by Oyoyumi. Most of the element has transfer factor values less than 1, representing there is a reduced chance of translocation from soil to plant. However, transfer factor for arsenic was greater than 1 in 100% farm soil and mixture. We can see as the rest portion increase, the transfer factor value decrease in all 3 crops, indicating raised material is contributing to dilute the arsenic concentration in the treatments. This table shows the health risk index for carrot biomass in male and female. Here, health risk index is less than 1, representing the human population is safe to consume carrot biomass grown in soil amended with raised sediments. Lettuce and tomatoes also have similar trend. We also observe a similar trend of arsenic with an HRA value higher than 1 from the transfer factor of translocation data. So we can conclude that it's not only coming from trace material, the background level consistently contains arsenic. These indices provides a comprehensive view of potential risk and allow for better management and mitigation of heavy metal contaminations in the environment. So from the previous research work and my current work has shown successfully that the trace material can be used for growing crops. There is less than heavy metal uptake for health risk when farm soil is amended with trace material. This indices provides a comprehensive view of potential risk and allow for better management. And by lowering, if you can use trace material, by lowering the need of chemical fertilizer, the trace sediments enhances plant and soil health, promoting the agricultural sustainability. Um, these are some of the references that I use in the slides and in my study. And finally, I would like to thank my advisors, Dr. Jawi Su and Dr. Angelica Vasquez Ortega. I would also like to thank all my lab members and some of my professors in my department. I would also like to thank the funding agencies, the Ohio Street Grant and the Ohio Environmental Protection Agencies. And I'd like to thank you all for listening to this presentation. Thank you so much. If you have any questions, I'm here to answer. Thank you.